This is the review of the DNA notes that we have covered already. So if you don't have these notes from before the break, they are found in, excuse me, in Canvas. And there's a link to them for Google Classroom for you to get the completed notes for you to use to complete assignments this week. I'm just going to review here, and I'm going to apologize if y'all can hear music in the background. My neighbors have extremely loud music going on, but it's been going on all day. So I'm going to kind of go with it here. Hopefully it's not too bad. This is a molecule of DNA. Remember, this is the background stuff. So what is DNA? Not what is the structure, just the background of DNA. We know it's found in the cytoplasm of prokaryotes, and in us eukaryotes, it's found in the nucleus. The DNA stands for deoxyribonucleic acid, and DNA is arranged in units called chromosomes. A chromosome has thousands of genes on it, and a gene carries the directions to make one protein. So, for example, our skin tone is that way because the DNA in our cells tells us how dark or light our skin's going to be, and that is how much protein called melanin, which is made. The more melanin you have in your skin, the darker you are. The less melanin you have, the lighter you are. So every gene codes to make one protein. And remember, from the macromolecule unit, we know that proteins make up all of our organs in our body, and they make up blood. So the chemical structure of DNA, that is found right here. So cracking the DNA code. Any questions that ask you what is a nucleotide, what are what types of bases are in DNA, what kind of bonds are in DNA, what's a nucleotide, all of that stuff is found right here. This is a nucleotide. It's a phosphate, a sugar, and a nitrogen base, which is right here. That is the building block of a nucleic acid. They both begin with nucle, and that C is a hard K sound. It's nucleotide, not nucleotide. So it's nucleotide. This is one artist, and this is a different artist. So the orange circle here is colored green in this other diagram. The pentagon is the deoxyribose sugar right here, and in this diagram it's blue. And then the squares are all of your nitrogen bases, and there's 4 types. So adenine is A, thymine is T, cytosine is C, and guanine is G. So those are your bases, and the hydrogen bonds hold the bases together. So these little dot lines are hydrogen bonds, and your A and T always bond, and then your G and C always bond. And this is where you're first told what a codon is. So cracking the code, how to read the DNA. A codon is 3 nucleotides long. So right here, TCA is 1 codon, not including the nucleotides, which is the nitrogen bases and the sugars and the phosphates. So all of this right here is 1 codon. So what is the chemical structure of DNA? That's here. Now, what's not written here that's in your notes, the backbone of DNA. The backbone is the sugar and the phosphate. So it's like the outside rungs of the ladder. Every single life form on Earth has our DNA alters sugar, phosphate, sugar, phosphate, sugar, phosphate on the outsides. Here's phosphate, sugar, phosphate, sugar, phosphate, sugar. What is different for all of us is the middle, the order of the bases. That is the code. So this particular code, this AGTC, this could be part of a code that makes you have black hair. And if you have blonde hair, you may have all G's here, G, 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 and G. So the order of the nitrogen bases is the code. Okay, here's where we practiced complementary means opposite. So everywhere you see an A T bonds. Everywhere you see a G and a C bonds. So if A is here in black, in red it bonds with a T. So everywhere you see a T, it bonds with an A. So they always bond together, and the C's and the G's always bond together. And we all y'all knew how to do this before the break, okay? Because this was the Monday, this was actually done the Friday before the break, the week before our break. Okay, DNA replication. Here's where you see complementary means opposite, and this is how it replicates. Basically, in steps 1 and 2, the DNA unzips. So this dark colored backbone, that's the original DNA. The light colored, the clear colored one over here, these are the new strands. So the original strand unzips, those hydrogen bonds break, and then new DNA nucleotides are added. And we know from the mitosis unit that in the cell cycle, this occurs during S phase. So you have G1, S, and G2. So S phase is when the DNA is copied. Okay, now RNA. So when questions ask you about RNA, start here. RNA stands for ribonucleic acid. This is what it looks like. And then these are our differences. You don't see a T here, you see a U. The sugar is down here. So here's your nucleotide or building block of RNA. It's still a nucleic acid, so it still has a sugar, a phosphate, and a nitrogen base. The difference is the sugar is ribose now. So it's ribonucleic acid, not deoxyribonucleic acid. And if you remember, the D means without, oxy is short for oxygen. So this is the sugar ribose, this right here is a sugar ribose, and it actually has the oxygen that deoxyribose is missing. And we talked about that in class. So this is a single-stranded molecule, and this is mRNA, or the messenger. Okay, so this one, when it's asking you about RNA, continuing on, here are your 4 bases in RNA. So if any of your questions ask you about RNA, you start here. These are the basics. A and U bond going down in here, A and U bond, and G and C bond. Transcription right here. That's when you use DNA to make mRNA. So this is mRNA. The mRNA is the DNA code, but mRNA is single-stranded, so it's skinny enough to get out of the nucleus. DNA is double-stranded, right here, and it's too fat to get out of the nucleus. The little pores in the nucleus are wide enough to let RNA out, not DNA. So DNA can't fit. It's forced to stay in the nucleus. Now, we ended up by talking about how to read the codon chart. So here's the word codon defined again, but remember it is in the DNA basics notes. But you're going to look up, so remember you have this filled out in the notes already, AUG. You have the first letter in the codon chart goes down here. So we're going to look for A, that's this part right here. The second letter is a U. So the second letter is one of these 4. And so that's a U. So now I have A and U. Where A and U intersect are these 4 words. Then the last letter in the codon is a G. Now, the third letter, this is where some students get tricked up. There's a G right here. Here's a G right here. Here's one right here. And here's one down here. So how do we know which G to use? Okay, well, the first letter is an A. So the third letter that we use corresponds with this A. So that means it's only one of these 4 letters. That means we're using this G here, not this G down here. This G down here corresponds to the first letter in the codon being a G. This G right here corresponds to the first letter in the codon being an A. And that's what we have, AUG. So AUG, here's our G. I move over and you get the word methionine, which also means start. So methionine also means start. if we look up GUC, and again, we've done this already, GUC, the first letter, I come down, there's the G. So I'm down here at the bottom. The second letter in the codon is a U. Well, a U is the first, there's the first letter. See, that's the second letter, and I have these 4 letters, and it's a U. So if you notice, does it really matter what the third letter is? If you thought no, you'd be right. All of these words are identical. Valine. Okay, now GCC. We're here, G again. The second letter this time is a C right here. Now we're right here. Well, all those words are the same thing too. That's alanine. Let's look up CGA. CGA. So the first letter is a C, so I'm right here. I'm at this second line right in here. The second codon letter in the codon is a G, so I'm right here. So where the C and the G combine, they meet, is right here. It can only be 2 words, arginine or, actually arginine is it. So that's the word. But let's look at that third letter. It's CGA. Why am I not going to use this A? Okay, this A corresponds to the first letter in the codon being a U. And our codon is CGA. So we have to use the A that's in this line. So that's right here. That's this A here. And that's going to be arginine. Now, today, your type of RNA, you have a picture identification formative assessment. And I actually have this at the top of that document for you to look at, especially for those of you that are having to toggle on a cell phone between notes and doing the Google form assignment, especially if you've lost your notes or you left them at school, or so you're going to kind of have to go back and forth. So to help those of you that had to do that, I just went ahead and took this picture and put it at the top. So this is your messenger RNA, and it carries the directions to make a protein. This is the ribosome, rRNA. So the little r stands for ribosomal. Don't worry about the AL at the end. That's just grammar. Okay, the root word of this is ribosome. Okay, the AL again is just grammar. So ribosome RNA is not grammatically correct. It's ribosomal RNA. So that's your ribosome. That's where the proteins are made. So this mRNA squeezes it out of the nucleus and goes and lands on a ribosome. The ribosome makes the protein. The transfer, or tRNA, it transfers from this end, which is the anticodon. That is nucleic acid language, AAC, GUA, AUG. It transfers from nucleic acid language to amino acid language. Okay, so amino acid. And here's your amino acid. Okay, so your classwork for today is like an identification, just to help you identify from various pictures from different artists get a feel for what the RNAs are going to look like. It's kind of like a doctor. You don't want the doctor to operate on you for the very first time he or she's ever done an operation. They're going to have practiced first. And they practice in med school on cadavers. And then they go through their residency where they get a lot of practice before they do the surgery, where they're the person that's in control and they're the main surgeon. So you want them to practice. So we also want them to practice as well. You are going to practice looking at different artist diagrams at actually looking at identifying the types of RNA. Okay, tomorrow we will actually go into this, will be Tuesday's lesson. Okay, so this will hopefully help you out to refresh your memory. And again, you can always, the very first hour, Monday through Thursday that we're going to be meeting, the very first hour you can get on the Zoom class and you can ask a question. Hey, Ms. Edwards, I don't get this. And then both Ms. Zahn and I will be on those Zoom meetings, so you can ask us questions. Okay, you can also type in questions also, and the both of us are going to be kind of sharing that responsibility. So if you have questions, please ask. Okay, and we're working on this together, so take care, and we'll be talking to you soon.